Hello and welcome to my channel, Sam Biology Class. Friends, today we will discuss about the principle of dark field microscopy, its advantages and disadvantages. But before that, will you please consider subscribing my YouTube channel? Your subscription and viewership are often a lot to me. Dark field microscopy is a light microscopy technique that is used for the direct observation of live, transparent biological samples without the need for fixation or staining. In this technique, the specimen appears bright against a dark background. Hence, the name is dark field microscopy. This technique utilizes transmitted white light from a specific source and a specialized dark field condenser which transmits a hollow cone of light that strikes the specimen from sides. Key components of a dark field microscope are light source, a specialized dark field condenser, objective and eyepiece. The source of light can be a halogen or a LED lamp. When the source transmits white light, because of the presence of this dark field stop at the bottom of the condenser lens, direct light is prevented from entering into the condenser lens. Through this dark field stop, only the annular rays enters into the condenser lens, which then produces a hollow cone of light. This is the hollow cone of light that hits the specimen from all sides. When the light strikes the specimen, it is diffracted, refracted or scattered by the specimen's edges, boundaries and its internal structure. Only the light that is scattered by the specimen is allowed to enter into the objective lens that result in a high contrast image even if the specimen is transparent. In dark field microscopy, only the light which is scattered by the specimen is allowed to enter into the objective lens, which forms the real, magnified and inverted image, which is then further magnified by the eyepiece to form the final upright and virtual image, which is visible to our eye. In this technique, only the light that is scattered by the specimen is allowed to enter into the objective lens. Remaining all light are prevented from entering. As a result, the specimen appears bright against a dark background. In this technique, the dark field stop, which is present at the bottom of the condenser lens, prevent the passage of direct light through the specimen into the objective lens. As a result, the background appears dark. On the other hand, the specimen which scatters light appear bright. This technique is highly useful for direct observation of live, transparent biological samples such as bacteria, other microorganisms, cells and tissues. Dark field microscope allows for the direct observation of living unstained organisms. This technique avoids artifacts that will be caused due to staining. Number 2, this technique produces high contrast image of transparent specimen, making them appear bright against a dark background. In addition to this technique offers superior visualization of thin transparent specimen or small sample that will be invisible or barely visible under bright field microscope. Disadvantages: dark field microscopy requires high intensity lighting that can cause significant damage or death to the live specimen. Number 2, this technique is sensitive to dust particles and other impurities in the sample which are highly visible can mimic the specimen. Number 3, dark field microscopy requires very clear, thin and wet specimen. Dense samples can inhibit light scattering, leading to poor image quality. Finally, this technique provides limited internal details. So friends, this is all about the principle, advantages and disadvantages of dark field microscopy. For more updates, subscribe my YouTube channel, Sa Biology Class.